afternoon and welcome to Pasco Laboratories. Today, I am Roger Palmer. I'm product manager for Pasco. And we're going to take a look. If you look at the materials here on the desk, I know that I'm probably the last thing you're going to do before you head home to cook some dinner. And you may think that's what I'm starting with all this material that's here right now. But let's get started so that we can get you on to the rest of the day. I'm excited that we can show today some ideas about fats, oils, and what makes some healthy choices, at least based on assumptions from what we hear and recommendations from food specialists. Now, we're going to use the UV spectrometer to take a look at those because if you look at most of these, they look clear. You can see through them. Now, you may also see some color, which is great, but most of what we want to notice is outside of what we can see visibly, and so that's where the UV-Vis spectrometer is going to come in handy. First of all, let's talk a little about oils and fats that we have. Fats as a food source are quite rich in energy, 9 calories per gram consumed. Really a fat, as you can see on the left of this slide, is a combination of a triglyceride. Now that may look familiar if you just take just at the oxygens on the left side, split those from the double bonded oxygen. And you say, well, that looks like a carbohydrate, a sugar, a C with OHs on it, okay, carbohydrate. And you'd be right, it's about a half of a sugar. It's C with 3 carbons as opposed to C6H12O6. But what's hanging on to those oxygens is a fatty acid. Fatty acids are great, and the body can break both those sides down, the sugar part and the fatty acids, and so that's why there's a lot more energy in those systems. The other macronutrients, the big nutrients that we can face, are proteins, obviously, and that's just a long string of amino acids, so it's an amino acid polymer. But that only has 4 calories per gram. And the last one are sugars, carbohydrates. Simple sugar, something like lactose or something, might be shown in that Fisher diagram there on the right. But when those circle together and then make a long chain, those become carbohydrates. And you may have heard of carb loading or something for having the energy available so you eat comfortably before an event so that the energy is available so that when you're out running it, you would have a ready supply because you're breaking those down. Something like pasta would have a long chain of sugars, and that then would be broken down to be CO2 and water vapor, and you get the energy. Now, let's take a look in terms of those fats. Those come from various sources. You may think, well, fats, absolutely. If you cook something that has fat in it, like bacon, it will have a layer of liquid there because it's warm. It's a liquid phase. But if you let that cool down, it'll become congealed in a solid. Whereas those that I'm listing are mostly, as you see from the store, and I have listed or shown in front of me, liquids at room temperature. Sunflower, the nut or the inside of that flour. Same thing with canola, but it's much, much smaller. Those can be gathered together and pressed so that the oil can come out. Might be something like a peanut in that next example. Even fruits can have oils in them. You can see that there's avocado, and that can be crushed and the oil separated, or olives, and that oil can be extracted. Fish can be ground, and then slowly the separation to bring the oils out of that meat. Or back to something like at the bottom where there's a sesame seed. All of those are listed and shown here in front of me. I think that what's fascinating is what is different between those and the fats, the solids that we see, the animal products. And you may say, well, is it really just all animals that make the solid? Here is a good example. I'll say no. Here's a good example of a plant. Can hold this up and I'll sort of see. This is from a coconut. And a coconut will have, as one of the legs of that acid, you can see that's definitely solid. Looks a lot like bacon grease. Because if we look at the molecules, palmitic fatty acid is part of that chain. Here's that chain in a 3D sort of model. You can see if they're all single bonded, look at how well they sort of line up. As a matter of fact, this molecules and the next molecules hanging down with their branches would line up quite nicely. And we know there always is some form of attraction, like London forces, the sort of instantaneous dipole that happens because the electron moves around. And so these will be attracted to each other enough so that even at room temperature, they'll get to stick together. You have to warm them up before they become a liquid. So palm oil would be having branches that come down the sides of this triglyceride, and that would also line up. All single bond. It's a 16 carbon chain. But animals usually have these 18 carbon chains or even longer. Stearic acid, for instance, as the side chains. The longer those are, the more the London forces, again, encouraging them to be solids. So what happens as you look down below those? Those are about where you start to see the side chains on this. Okay, here's that fat again. Start to form. I'm going to pull one of these off right here. Let's pull this guy off so we can see. Here's one fatty acid. This is the carboxyl at the end. But if instead of at a single bond at every carbon, let's take a look and make a double bond. And notice what I'm going to have to do. If this was a hydrogen and this was a hydrogen, we're going to have to take those hydrogens off. And instead of being saturated with hydrogen, now we're going to be unsaturated. And instead, you're going to hook two bonds with the carbon, between the carbon. So it doesn't have all the hydrogens possible. That's why it becomes unsaturated. What's interesting is the double bond becomes more reactive, and your body can do chemistry with that to get rid of it, to burn it for energy, to work it through your body. And that's what makes them more healthy. It's easier to deal with inside your body. So you're starting to see olive oil would be a 18 carbon chain. It's got mostly oleic acid. So of those fats that are in it, at least one of the chains has a longer chain in it. Now, what happens is that all these other bonds can rotate and move around, and so they're, as a matter of fact, kind of line up. Once you put a double bond in, that starts to bend it, and it has to stay bent. And so when these try to line up, there's a kink in that system, and they sort of stick out. And if there's more than one double bond, there might be two. So one of the legs of this three kind of armed molecule starts to really stand out, and it can't line up like it did before, and so they stay liquids like we see. As a matter of fact, if you look down below those, you start to see 2 or 3, 4 double bonds, and sometimes it's easier to count from the end. And so you may hear from nutritionists say, oh, that's an omega-3 or an omega-6. What they're doing is counting from the other end, from the functional group. If you've had your early organic chemistry, you usually know that you're supposed to count from the functional group on the carbon chain backwards to show where some function is. Well, in the nutrition world, they start from the omega side, the end, and work their way back. So when you see something like safflower oil, it's 18 carbons, but you start to count from the end, and you'll go in 6 carbons before the first one. That's an omega-6 or an omega-9. Okay, so you may hear combinations, omega-3 and omega-9. And so people will make claims about those, but that's all they're doing is counting where the double bond is from the end of the chain. So all of that's great, and you'll see most of those vegetables have a double, 2 doubles, 3 doubles. We'll call those polyunsaturated bonds, and that's what we're saying is, in health sort of circles, is what is good for us. So we want to see of these oils, which of those that are liquids are the best and healthy. We're going to assume they're unsaturated, having more double bonds. So what we're going to do is we're going to take our spectrometer here. I'll go ahead and turn that on. One of the things that we will want to do is to calibrate it so that it knows what the normal amount of light should look like. Okay, we're sending, I'll show you the spectrometer again here. We're going to turn on a light source, which is very warm. It's a hydrogen sort of incandescent bulb. A fluorescent bulb that we shock and send, like a hydrogen lamp or a mercury lamp, or mercury is going to be in your room. Spark through there, and then those electrons, as they fall back, are going to give off light in the ultraviolet. Hydrogen's easy to go through there, so it's easy to excite, and then that light that comes off is mostly and it's actually deuterium. It's a special form of hydrogen, but that particularly gives off light in the ultraviolet. We also have a nice wire lamp, just like your light bulb that you'd screw in would be there. And that gives us the visible and the infrared. So this spectrometer can see both in the ultraviolet, the color region, and the infrared. It's got a light source here, and it's shining that direction. Your sample goes in the middle, and then the reading of this is going to be on the other side, where they are clearly shown on this face of the printed spectrometer. So now, the first thing we want to do is say, hey, what does normal look like? What does it look like when there's no compound in the way? So we're going to take whatever solvent that you needed to dissolve your substance. In this case, it could be like hexanes, because hexanes look a lot like 6, 8 carbons in a row, and you could put that in the pathway. And what I'm going to do is get out of the presentation. We'll open up a version of our software, which I'll have my assistant put into, friend and colleague put into the chat. So if you're looking for it, and it's called Spectrometry. So we're going to, well, you know what, let me just show you what it looks like on my desktop. It can be downloaded and put onto any kind of machine. I will go ahead and open this one up so that you can see what it looks like when it's first started. Put it on a Mac, you can put it on PC. It can be run on an iPad, on a phone. It's small geography, but it can work. You can also transfer files, like to, for instance, there we go. I'm trying to think of the Android notebooks, so that while it doesn't have USB that could hook to this, you could still get the data to do the analysis. So all of these will run the spectrometry software, and here's what you're seeing. And the software, I'm going to switch to say, let's do these two buttons on the bottom, which is to calibrate the light source. So we're going to shine light through that substance. The detector is going to say, that's what full light looks like. We're going to also shine through when there's no light that's coming through the spectrometer. And we're going to say, that's what dark looks like. And then what's missing, what's different than that, is going to be what the spectra do to that substance, like these oils that we're looking at. All right, let me run those real quick. I'm going to run with no light in there, and the spectrometer knows what no light should be, what the current coming off that detector is. I'm going to run it with the light shining through that substance, and it closes back again. Now it's ready to go. I have tools on the bottom that I can spread out that spectrometer, and I'm going to start recording. We'll take out our clear sample, and we're going to put one of these samples in. Nope, let's put it in the right way. Now, the glass materials that we offer, the cuvettes, are made of special material. It's not glass. If you use glass, it's great for visible light, but it blocks UV. So this is a special plastic. It's also that there are special glass, which is expensive, called quartz. The quartz would allow the UV light to go through, but there is also, for schools particularly, it's great because they call it disposable UV cuvettes, and it would be a great, much less cost to use those. I'm going to put those in. Now, what's happening is these thin ones have a much smaller volume on the bottom, so that if you're doing things like organic chemistry and you don't produce a lot of material, it's a much smaller volume to work with and shine through. And that's more appropriate for, smaller amounts like you'd normally see in a lab. I've just put that in, and we're going to run the spectrometer, and just to see, we have a spectra that comes out, an absorption in the ultraviolet. I'm going to go ahead and make that bigger, fill the area. You may say, oh, that looks a little bit noisy. It's got some edges, but I can smooth that down as much as I want to give what I would expect to be a normal spectra. We could take a number of scans and make that look a little more calm. That looks great, and I will go ahead and stop. What's nice about this spectra is that where that peak occurs is dependent on what this molecule is, contains in it. We were showing these lines. Anytime you have a double bond, for instance, the electrons get excited about ultraviolet light because they will change and they can absorb that light, jump from a bond that's between these carbons to an antibond. That's the right amount of jump for ultraviolet light. Now, what's interesting is we're going to take a look at several more of these in just a second, and I'll talk more about that. But let me just say, can we use the software to measure that peak? And I'll say absolutely yes. The tools are down here at the bottom. I can put a marker. Notice as I get close, I'm going to say, yep, that is easy to put right at the peak, and the software will tell me what that peak value is. In this case, 297, right around 300. Okay, that's great. What I'm going to do is drop out of this. It's sort of a Julia Child, and since you've seen one process, I'm going to say that's great. You could do that with all the rest of yours. Let's go back into the presentation that I've got so that we can see what it would be like to do all of these substances that I have in front of me. Okay? After running all of these today, I see I've got the different oils that are in front of me. As a matter of fact, here's my sunflower, which is the lowest on the list, and then the peanut oil. Matter of fact, they're in the same order as I have them on the table. All right? We're working our way down to get to eventually the olive oil. And then I had two that I just had very small samples of, which were great because I was interested, like fish oil. So if we take a look at these, you're going to notice that those spectra move further and further to the right. The more double bonds that I have in one of these side chains, the more of those that happen, they do influence each other in terms of what energy can excite or move the electron from bonding to antibonding. And so they become less energy to jump because the bands become closer. It takes less energy for those electrons to go from the bonding orbital to the antibonding orbital. What's nice about that is we can use that phenomena to our advantage to determine if something is healthier, well, what dietitians would say are healthier, and that's the unsaturated. The more double bonds would move to the right because it takes less of a jump. So more lower energy or higher wavelength light can make that excitation happen. What's going to happen is as you move across, you're going to get to these oils that are eventually, like we have a list here of the olive oil. And I'm going to follow that one. That's sort of in the middle, or the avocado oil. You might see that there's sort of a little bit of some disturbance in the blue and a little bit of disturbance in the red. That's typical of photosynthesis. So there is definitely some chloroplasts that are in some of these oils that are over here on this side. So we're seeing that as well. So we have to kind of, I don't want to say ignore it, but we say, okay, that's there too. And you certainly could study that process and say how much each different version of olive oil or avocado oil that's out there. But let's look at the general pattern back at the main absorption on the left. You'll see that they slowly creep to the right as more and more double bonds happen. So we can say, for instance, that sesame oil has more double bonds because it absorbs all the way down into this sort of further right area, or fish oil, or the avocado oil. Now, you may say, okay, that's great. Those are those more healthy oils because they have more double bonds. They're more unsaturated. If you look just across this list, you're going to notice they have sort of more and more color as we move to this direction. And I want to make a point about that. Here's the color wheel that's out there. If you look at what color something is, for instance, let's do a comparison to something else, and we'll back apply this to these colors of the oil. This is a blue substance with some blue food coloring. Now, the reason it's blue is because everything but blue has been more absorbed. Only the blue is making it through. So looking at the blue in the color wheel, that means its opposite side of the color wheel is being absorbed, the orange. Okay, so if orange is being absorbed, that means the blue light's coming through. So if the yellow and green are being absorbed by these lights, by these oils over here on the left, the avocado oil, the olive oil, what is opposite the yellow greenish border? It's more of the violet. So yes, it's starting to absorb more violet because there's more double bonds, and what's coming through is that yellow. So that's an easy way in terms of your visual sense to know also oils that have more double bonds. Let's take that understanding and sort of see in across food sections after doing that sort of analysis. If you look at the first column, red is everything as single bonds or a saturated condition, like meats, bacon, grease, creams, that kind of thing. I apologize, that's the middle. Everything that has more double bonds, like oils and nuts, are going to be represented, that's here, and they have very little of the red, very little saturated. Doesn't mean every side chain on here is got double bonds. It just means more than some others. Remember, these can all be various side chains. They don't all have to be the same, but they have side chains in general. If you look at more of fats that come from animals, butter, chicken fat, maybe bacon grease, dressing, those kinds of things. Obviously, dressing is more likely to be a vegetable oil. They are starting to show more red. The dairy products, highly animal sourced, are mostly single bonded, and they're solids, just like bacon grease or turkey breast, which has fat, salmon that has fat in it. All of those are high in saturated fats, okay? Just because they're meats. Now, you can choose some meats that are lower in terms of health, and that's great. But in general, as we look across those, that's what the difference is between these sources of fats, which we need in our diet. I'm not saying we can't. We do need those, but we can be mindful about how much is saturated, which is supposedly less healthy, and it reported as less healthy in those that have double bonds, which is more healthy. So All of this was possible to study in more detail because we had a spectrometer. The UV-Vis spectrometer that we are using here is a great example of what you can use in your organic chemistry class. Organic chemistry would possibly need some great precision, which if you look under our resolutions, we can differentiate between about a nanometer and a half peaks that occur. That it spans the gap from 180 nanometers to 1050. And if you're looking at the light source in the upper right, that's UVC, UV in the B range, and UVA. All of those are well covered by our spectrometer. You need special cuvettes to see that far down, and that's why we're offering the cuvettes that we have that are disposable, or those expensive that if you need the quartz to go well into the UV, we have those as well. And lastly, the sort of the range and the accuracy, but also our a warranty is 5 years on all the projects that we do make, like a spectrometer, except for the light source, which of course those have a lifespan. We do cover it for a year, and good use, about 1,000 hours is typical under that category, but we just call that a 1-year warranty on the bulbs, and those are replaceable and available. Now, the sensor that detects the light on this side is a CMOS detector. It's just a newer form. If you're a photography sort of into photography, you know the CMOS. Every cell reports out instantly, and then we write immediately to a database. It just makes it a little faster in terms of its writing, which is great because it's scanning every second, generating 2048 sort of bits of data every time it scans. We offer the quartz fibers. If you want to do what light is happening outside of the spectrometer, you can then funnel from outside the spectrometer back into the spectrometer. And so if some light sources out here that you want to monitor, like a flame test or something, that you can funnel the light in. Or if you have something that's big, you could have a great UV source shine through your larger outer, outside of the spectrometer into this, and then you can analyze the light there. We also sell, like you see, the replacement bulbs on the inside, any of the UV cuvettes. They can come in 10 packs or 100 if you're just trying to keep a lab supplied. So all of those things are wonderful. You may wonder what's the normal size, especially if you're doing large freshman chemistry where it's a solution that's not terribly expensive. We sell those that is a 4.5 mil cuvette. It's fully square. But you might notice on that new diagram at the bottom there, what's called semi-micro. If you're doing organic chemistry and you come up with, just less than a gram of your material and you want to be able to still do an analysis, semi-micro is great because at the bottom is 1/3 of that width is all that you need for the liquid. And in terms of the whole cuvette, it's 1/6 of the normal volume that you need. But you can also use your own if you've got micro cuvettes and you're doing DNA analysis or things like that, which are visible in the UV. So if you have your own micro pipette supplies, you can order those, but the other two we make available so that you can do this analysis. Spectrometry is important, is what we're saying. We saw that we could see things like saturation and unsaturation, but in addition to seeing what the molecules are made of, like we did in this oil example, we can also find out what concentration of substance is there. The height of that peak tells you the concentration of the substance. If you want to do a quantitative analysis, like Beer's law, then we can show something that has if you create solutions that have various concentrations, now you can find an unknown concentration. You can follow how fast those change in case a reaction happens and the thing you're monitoring is going away or appearing. You can monitor that. And the last one is you can use our quartz cable to monitor light that occurs outside of the spectrometer itself. The labs that you can do, we want to be able to assure you that our support is there for you. These are the most common ones that we know are happening in terms of UV analysis, from unsaturated fats, aspirin generation, and seeing if that's aspirin or if it's the hydrolyzed sort of byproducts. You can tell the purity of what's been produced. Suntan lotions, in terms of why they work, because they're blocking UV, make good labs. And then we can look at light from outside sources just to determine some physical constants as well, if you're watching and are looking at those from the sort of understanding of the atom perspective, or if you're in physics and you want to understand the energies of orbitals. Those are all wonderful sort of tools we can do. And how did we do that? We started off our spectrometry software will allow you to look at what's in a solution. If you pick a wavelength, you can figure out the concentration you have, or if that concentration changes over time, or if you just have an outer light source, you can funnel that in. So the process in terms of doing it in the lab is first you calibrate the instrument with a light and a dark sort of the brightest light and darkest light. You put your known solutions in and finish with what you want to test, the unknown, which is you're using to try to find an unknown solution of concentration, like in this Beer's Law example. Maybe these lines that were here were collected at various concentrations. In the little call out you see in the upper window here is the spectra, and you've picked a particular wavelength where you know there's a peak. And then as you put several of these cuvettes in that have less and less of the material, you can determine what that looks like based on that absorbance. And you can then put in your unknown and find on that line that goes through those what your natural substance is. In this case, you see there's an extra green sort of circle, and that looks like in terms of concentration is about 0.35 of absorbance. And then if you read straight down, you could read what that concentration is. If you want to follow concentrations of a reaction that's changing inside the cuvette, you put those in and maybe that color is disappearing or that absorption is disappearing. So if we watch two substances going through a bleaching process or losing some part that absorbs UV or visible or infrared, you can see that one substance is slowly losing and becoming less and less visible. The light's getting less and less that makes it through. Or this second example, which is happening much faster, then you can do the kinetics. So with those kinds of ideas, not only can you set up an instrument quite easily, determine how light can show you concentrations, how fast reactions happen, but that you would be quickly on your way to doing some great science. If these techniques interest you particularly, we're going to spend more time in our next workshop, which you can register for at pasco.com/training and events. And if spectrometry is of interest to you, we do have a sort of a reading page, a white paper kind of page that talks about spectrometry, which we'll drop in the chat. So take a look at our future training events or browse our pages online. And if those are great and helpful for what you're doing in your science classes, please leverage those. In that case, we love doing science together with you, and we hope that you've learned a little bit more about where fats are at. We're looking forward to providing any kinds of help that we can in the whole field of spectrometry, understanding chemistry, and doing great science. With that in mind, we appreciate your time today and look forward to other ways that we can show you great science.